Hello everyone, let's begin the second class of Module 4. Here I'd like to talk about seed germination. Seed germination is the process by which a seed begins to grow into a new plant. Cannabis plants are usually propagated by sowing seeds in seed beds. If seed has been germinated and sprouted, is referred to as a seedling. A seedling about 5 to 6 inches tall, it will be transplanted to a larger pot later. Seed germination is the metabolic processes leading to resumed growth and develop into a seedling. The nutrients needed for germination in the ground are supplied from the cotyledons, and when the sprouts rise above the ground, the newly formed leaves provide nutrients to the plant through photosynthesis. At the beginning stage of germination, seeds take up water and swell, and the seed coats and the pericarps soften. At the second stage, stored substances are broken down by enzymes. Then radicle, the first root, breaks through the seed coat and the pericarp and grows downward. After that, the shoot grows and forms the true leaves. This slide shows the several growth steps of germinated seedlings. If germination occurred in an environment outside of the soil, it's best to carefully transfer the young seedlings to a starter. They can be grown in the starter tray and transplanted to other containers or pots. Seed germination can take 12 hours for a maximum of 3 weeks, depending on the seed structure and germination conditions. The first stage takes 12 hours to 3 days after sowing. The second stage usually takes 3 to 5 days, and the third stage takes 7 to 10 days after sowing. This photo indicates the developmental process of germination. During 12 to 24 hours, the cannabis embryo absorbs water until the radicle breaks through the seed coat. In 24 to 48 hours for the development of a radicle. The primary root grows very rapidly under normal conditions. They may reach a length of 7.5 to 10 centimeter within a period of 48 hours after emergence. In 48 to 96 hours, cotyledons emergence and the development of 2 fully open cotyledons. There are 2 types of seed germination, such as apigeal and hypogeal germination. Cannabis shows apigeal germination type. Its characteristic is that cotyledons are pushed above the soil surface when the seed germinates. Cannabis seed germination rate declines quickly with storage time, especially under warm and humid conditions. The germination rate of cannabis seed is approximately 60 to 70% after 1 year, 30 to 50% after 2 years, and drops to below 20% thereafter. Next, I will talk about the germination method. There are usually 3 methods that are commonly used. The paper tower method is the easiest way to germinate the seeds. Wet 2 moist paper towels, making sure they are damp but not soaking. Place 1 damp paper towel on a flat surface. Arrange the cannabis seeds evenly on the damp paper towel with full contact. Cover the seeds with the second moist paper towel and fold loosely. Place the paper towels in a Ziploc bag to maintain moisture and create a humidity dome effect. Second, a direct to soil method is that soil should be prepared. It must be loose, well drained, and well aerated. A light, loamy soil with organic matter is ideal. The planting site should be moist but not waterlogged. Seeds are planted at the depth of 0.4 to 0.8 inches. The important thing is to place the seed pointed end downward as shown in the slide. Gently cover the seed with soil, and the soil should be lightly watered to ensure good seed-soil contact. The final method is using starter cubes. Starter cubes give the ideal environment for cannabis seed germination, especially in hydroponic setups. Soak starter cubes in pH balanced water until fully saturated. Gently squeeze out excess water, leaving cubes moist but not dripping. Create a small hole in the center of each cube, about a 1/4 inch deep. Sow 1 seed per cube in the hole and place into slots in the tray with the dome for the humidity. The important point is to plant the seed with the pointed end facing downwards. The left picture shows a seed that was planted upside down, causing the roots to grow upwards outside of the starter. As you can see in the picture on the right, the pointed part is where the pericarp opens. Essential conditions for germination include moisture, oxygen, and darkness at 65 to 77 degrees Fahrenheit. The seed consists of 2 cotyledons and 1 radicle. 2 cotyledons store a large amount of protein and oil. These nutrients are broken down by various enzymes during germination and used as an energy source. As shown in the picture on the left, the cells of the cotyledon are filled with large protein bodies and smaller oil bodies. The protein bodies are surrounded by numerous small oil bodies. A protein body is composed of a central crystalloid and the peripheral protein mass. And most of the enzymes, such as lipase and other hydrolases, are existed in this matrix. In the germination of cannabis seeds, the stored oil in the cotyledons play a significant role. In the germination of cannabis seeds, the stored substances are broken down into the low molecular substances such as amino acids and fatty acids. Also, fatty acids are converted into sugar through the glyoxylate cycle and then moved to other locations. Eventually, the seed grows into a young seedling. In this unit, we look at the seed germination of cannabis. All right, everyone. That's it for today. Thank you. Bye.